So, here we go. I thought I could say something about this because I have actually started a bunch of research initiatives in my career so far. These are some examples of them. And the latest one is called Sea Spirit, Center for Sustainable Plant Innovation and Resilience through International Teamwork. And I'm going to talk a little bit about how we got that going. But before that, I will also talk about a project that's not a center, but it's a research project that we've been working on in the last few years in my lab, which is going to be the subject of my scientific talk later. So I wanted to share with you in this part of the talk as a prelude to how we got started with that project. Kind of a back story. So I thought when Barno asked me to give this talk, what are some important things that I've learned from initiating projects and centers? And there were certainly things that didn't work. And I have to say there are a lot of things that I failed at. But the initiatives that did end up succeeding have all of these 5 things in common. One thing that's missing in the middle, of course, is funding. Forgot to add that. Kind of an important aspect. But all of the things that have succeeded, in my opinion, have all had very, very strong motivation at the beginning. The passion and the question. And then they also had the right people. And I think this is one of the most important things that I've learned in my career is it's really all about people. I went into science thinking that because I don't really like people, I'm an introvert, I don't like dealing with people, that, oh, being a scientist, working on plants is perfect. They don't talk back, they don't scream at you, and, you know. But it turns out that being a scientist is really about being a people person. And I've learned that the hard way. And I've kind of learned to overcome my fears of people. And I think having the right team is really the most important aspect. And then after that, you do need to get some funding to get the projects going. But once you have the funding and the people, really important things are establishing clear expectations and then having open channels of communications. And in order to do that, you have to really create safe psychological space. And this is something I'm still learning about. And I know a lot of excellent scientists and faculty still don't know how to do this well. And I think it's a big homework for all of us. So now I'm going to talk about the backdrop of this heat resilience that we've been studying in our lab in the last few years. And the motivation was actually very simple. I was living in California for a long time, actually, and we started having these heat waves that was just crazy. And then they ended up with big wildfires. And it was year after year after year, we hit records. And I could feel the heat, right? And everybody was really suffering a lot. These are some images of fires that we've had in the Bay Area. 1.5 years ago, Death Valley hit a record of reaching 56 degrees Celsius in the summer, which is enough to cook beef, actually. Rare. But still, there's some temperatures up there that show you how animals like us respond to high temperature. And I know this, Uzbekistan is pretty hot in the summer, and heat is not great for us. It's not great for any organisms. In fact, these are some examples of how in the middle, fruit flies die off in response to heat. I believe the red are the portions of flies that are still alive. So you can increase the temperature on the y-axis. And you can see like around 37, 38 degrees Celsius, most flies die. On the right is bacteria. You can see most of them crash around, I guess, mid-30s too. And then on the left here is photorespiration. Oh, sorry, it's photosynthesis and respiration of corn. And this is basically showing you that there's double whammy here. So respiration actually increases as temperature goes up and photosynthesis decreases. So the net photosynthesis is even affected more as temperature increases. So heat is bad news for almost all organisms, except there's some very few organisms that really love heat. And there are a couple of plants that are heat extremophiles. And this is one of them. This is a plant called Titisromia obliquifolia that grows really well in Death Valley. And this lonely plant in the front here is actually Titisromia. This is taken in winter. And then the day 130 are Titisromia growing in our growth chamber, mimicking Death Valley summer. The yellow part of the North American map shows you where it's found mostly. So an amazing thing about this plant is that in the middle of summer, it can increase its biomass by 30% a day, which is remarkable. And it thrives at temperatures like beyond 45 degrees Celsius. So it sounds really cool, right? It's not a model, it's not a crop, and so how do you even study that? Well, this is just to show you that even though it's not a crop, it's actually related to some important crops like sugar beet, spinach, quinoa, which you've heard about earlier, amaranthus. Well, we were the first ones to think about studying this. This was actually something that folks at Carnegie Institution for Science, which is an organization that I worked for before I moved to Michigan State, worked on this plant in the 70s, and they were pretty bold, I think. They basically put an RV with instruments that they built. These are before light core, so these are instruments that you could use to measure photosynthesis and respiration and so on. And these guys drove this RV around up and down the coast, the West Coast, and went into parks and places and measured photosynthesis plants. And when they went to that valley, they saw this plant and it did this crazy photosynthesis in the middle of the summer. They brought the plant back and created a greenhouse that would mimic that valley summer. I don't think they succeeded that well. I think this was a huge energy hog, and I don't think that research lasted that long. So anyway, it was something that went on for about 10 years at Carnegie. There weren't a lot of papers published on it, one science paper and then a bunch of Carnegie Yearbook articles. And then I heard, so the way I started working on this project is I heard Joe Barry, who's still at Carnegie, by the way, give a talk about all of the research that he's done throughout his entire career. And when he talked about this plant, I thought it was really interesting. And so I was curious about what kinds of biological innovations this cool plant had in order to survive or thrive under this extreme heat. And I wanted to study it from all these different angles: biochemistry, genomics, modeling, physiology, and so on. It's a kind of a big dream, I think, and to really study mechanisms of thermal tolerance. So the question is like, okay, well, if you have an interesting idea and you kind of got some, papers from the 70s and so on, how do you start that project? So, how do you find people? How do you even get the seeds? It's like you can't go to the stock center, right? How do you actually study them? Like, how can you mimic that valley condition, which is very unusual? And how do you do all of this to get funding and stuff? So I wanted to just share with you how I did it. I don't know how to look at, but it worked for me. So I think I was kind of lucky too in that I was able to get some seed money from Carnegie to hire postdoc, Corinne Prado. And the rest is pretty much history. Corinne was actually able to do most of these things in collaboration with another postdoc at a different department at Carnegie, which just kind of pro bono work with us. She was very interested in the project. She, in fact, was a postdoc in Joe Barry's lab, and so she wanted to just collaborate. And then we got a couple of volunteer summer interns. And then that's how we kind of built a team. And then once we started getting some results, a lot of people were just interested in collaborating no matter what. And these are some people in our lab. that volunteered their expertise. So we were able to do transcriptomics and genomics and so on. So we still hadn't gotten any funding at this point. I actually have to be honest, I still don't have federal funding on this project. But we actually built a lot of collaborators. So in many ways, like we didn't really need a lot of federal money to keep this project going because a lot of people were just interested in working together. But you still need the seeds. And so how do you get the seeds? We were lucky to find some botanical garden in Southern California that had these seeds of this plant. And we asked them for some seeds. They gave it to them and gave them to us, and we worked on them for a little bit. And then we asked them again because we ran out and we couldn't figure out how they would set seed in our greenhouses. And then we went back the third time, and they said no. We're not going to go back to the freezer anymore for you. You're going to have to go get it yourself. And so then we ended up going to that valley. You know, some colleagues, Karina went, Jen went, and I dragged my family on a vacation. And so this is my nephew and my mom who helped collect seeds. So we actually ended up collecting seeds ourselves. And we found two types, which was really interesting. So I'll talk about it the next talk, if there's time. Another thing that was a big bottleneck for us is to how to grow these plants at Carnegie. And so at the time, there wasn't a commercial growth chamber that could go up to the Death Valley summer conditions, which is shown in these two plots. The top one is the light intensity that we have to go up to 2,000 micromoles per meter square per second to mimic the summer of Death Valley. And then temperature had to go up to about 47 degrees Celsius. These are essentially the patterns of these parameters taken from the weather stations there. Anyway, we convinced this company called High Point to make us a chamber, and they did, but it broke every 2 weeks. And the reason is because it couldn't handle the heat and the light. And so they had to build us a unit that was as big as the growth chamber. And then we went into Bronson, which is a company, I think, in the Netherlands that took about 2 years to build this chamber, which is incredible. So we've been working with this chamber for a while now, but an opportunity arose. This is something that if you guys have an opportunity to move your lab or yourself somewhere, it's worth taking the risk because for me, moving to Michigan State University allowed us to actually get our 4th generation growth chambers from this company called BioChambers, and now we have 3 of them, each being much bigger than what we had before, and they are able to mimic the Death Valley conditions pretty well. And so Corinne basically says this changed her life because she no longer had to like babysit the growth chambers. So these are some of the things that we had to deal with, and some of the results that we got from studying this plan. There's time, I'll talk about it in the next talk. I want to end this part of the seminar by talking about this new project that we started at the beginning of this year, Sea Spirit, because I think that it has a lot of the elements that I talked about of being a successful initiative, even though it's only been about 6 months. I kind of feel like this is something that's gonna really take off. And again, the motivation is temperature, drought, pathogen load as a result of climate change and this food insecurity as a result of that. Most of you probably know this, but more than half of our calories come from only about 4 plants. This is wheat, rice, maize, and soybean. And all of them are very sensitive to high temperatures. So at around mid-30s degrees Celsius, the reproduction really pretty much crashes. Pollen's not great. And so we have pretty severe problems that are going to face us because this basically is a projection of increasing days of temperature being above 35 degrees Celsius. The blue dashed line is where we are today, and you can see it's going to continue to go up. And in addition to the increasing temperature, we are going to have an increased load of pests and pathogens. this is a really nice study that has interviewed farmers around the world. And they were asked, like, you know, what are your biggest problems? And it turns out the biggest problems of agriculture, especially for those food crops, is fungi, fungal pathogens, indicated by red here. Yellow, the insects, pretty bad for rice and maize, and then of course potato with phytophthora. So we lose about 20 to 30% of our yield of crops due to pests and pathogens on top of the heat. And then on top of that, we got drought, as Pharma mentioned. This part of the world is actually pretty vulnerable, as are many other parts. So these kind of triple, quadruple threats are something that we have to really think about. And this was the motivation for Seaspirit. And what we wanted to do was something different from traditional plant breeding and biotechnology. I know that a lot of you here are really interested in breeding biotechnology, which is great. This is actually, I don't want to go over all of these successes and advances that have been made through plant breeding and biotechnology. There's a review about this. I'm sorry about the format here, but this came out as a Darwin review recently. But you also know that breeding and biotechnology take a long time. Typical development period is about 5 years. If you're optimistic, and then there's another huge long regulation period of 10 to 20 years. And then on top of that, we've got public non-acceptance of GMOs and so on. So even though with genome editing like CRISPR, we can potentially get around the regulation issue, still takes a really long time to breed new varieties. Okay, and so what we wanted to do was to try to change the game a little bit. And this is another review I wrote with a bunch of people. This is in Trends in Planet Science about you know, thinking about changing our paradigm of going from lab to field, and thinking about doing research, fundamental research in the field, and then taking, you know, the observations or discoveries back into the lab to look at the mechanisms and so on. And so Seaspirit basically tries to find bioactive compounds from plants and microbes that would help plant resilience, especially in agricultural setting. And so we're hoping that this could bypass the hurdles that we face with traditional plant breeding. And we are trying to do this in collaboration with 4 other countries. Right now it's US, South Korea, Canada, Japan, and UK. But we're very interested in expanding this collaboration, maybe with Uzbekistan, maybe with Colombia. Vietnam is quite interested. And so, yeah, anyway, the plants that we're targeting here are 7 crops: corn, rice, soybean, potato, tomato, pennycress, and cassava. And we're focusing on looking at heat, drought, and fungal pathogens. I'm not going to really talk about how we're going to do this, but just to say that I'm going to skip over this, that there are probably hundreds, if not thousands, of regulatory metabolites, stress hormones that are out there that haven't been discovered. We already know, you know, that plants make a lot of compounds. We already know, we don't know the functions of most of them. And we only know about 9 hormones. That's not very many. These are the ones on top there. And they didn't necessarily all come from plants initially. They came from things like human urine or fish DNA. And then the compounds in the Rhee are some of the regulatory compounds that are behaving like plant hormones, but they're not really designated as such. And we think there's actually a lot more of these, and that's what we want to. And so this is sort of a QR code that takes you to the website. We have a project website, and we have 6 major aims here: to discover those compounds, to annotate them, find the genes that make those compounds, and then synthesize those compounds, test them in the lab in the field, and then test environmental toxicity and animal toxicity. So it's an initiative that feels much more like a startup company, but it has a lot of think potential impact for agriculture. And a lot of people in the project are really passionate about this. And so I kind of feel like we will get somewhere with it. And sorry about the formatting again, but already we have more than 80 people involved, and not all of these folks are getting money from it. So again, an example of something where if it's exciting enough, you're gonna get people to collaborate. I think that's the end of my talk, actually. So thanks for your patience.
Thank you, Steve, for the wonderful talk. So, are there any questions? Told it from? Career Jam. She's asked me.
Okay, there's a question.
Yes, she's interested in the project, and if this project also for the big students or if they read the
Yes, she mentioned, I'm assuming the Sea Spirit project. The second part, yeah, thanks for the question. I wanted to say that with Japan, we are receiving about 10 to 12 scholars per year. Not like huge amount of time, like each person's spending about 2 to 3 months, some people 6 months, some one person spent 2 years. So the funding is set up in Japan to send scholars to Michigan State to do research. And so I would love to expand that to Uzbekistan students and postdocs. We have started having a conversation with Vietnamese university. I think it could be a really great model. But it's very new, so we're still like developing how we're gonna work with each other, because it's actually not a huge amount of money, but it's actually very extensive with 5 countries being involved and every aim going in parallel. So we're still recruiting, people who are going to be leading the sub aims and so on. So the answer is, I would love to. I think that we would have to like figure out what might be the best mechanism of that. I mean, if we start publishing papers on this and publish that it comes out, I think a lot of countries, governments would be interested in getting on board. So, since I'm here, Barna's from here, we're making connections. I kind of feel like maybe in about a year or so, or 2 years, maybe you could expand this to Uzbekistan, or so on. Thanks for the question.
Yeah. Thank you.
So the answer was yes, but not right now.
I'll take that.
If not the case, we can